So we are going to resume with the microscopic anatomy of the bone. Our 5 major cell types that we are going to seek are osteogenic cells or osteoprogenitor cells, osteoblasts, osteocytes, bone lining cells, and osteoclasts. So our osteogenic are also called osteoprogenitor cells. So they are going to be stem cells that are actively dividing. We're going to find them in the periosteum covering the bone and on the endosteum on the inside of the bone. So when they are stimulated or called to action, they are going to differentiate into osteoblasts, so the bone building cells, and then some of them will remain as those stem cells. Osteoblasts, as you know, are bone-making cells, so they are going to secrete the unmineralized bone matrix, or the osteoid, so basically the scaffolding for the bone. So this osteoid is made up of collagen and calcium-binding proteins, okay? So collagen is going to make up about 90% of that, so a lot of resiliency, some toughness, but also the ability to bend and withstand stress without breaking. So those osteoblasts are highly mitotic. They're really working hard to make new bone. So then when they become completely surrounded by the matrix, like when they are done finishing making up the bone, they are going to turn into osteocytes, or the mature bone cells. So just a little look at what the osteoprogenitor cells look like, and then whenever they become osteoblasts, you can see they're a lot larger. They still have a single nuclei. So these osteocytes, so bone mature cells, they're sitting in their lacunae, or their little home. They're no longer dividing, but they are still alive and very active. So their job is to maintain that bone matrix and sense any stress or strain that the bone might be under. So its job is to respond to this, such as an increase in force, or the converse side of that, the weightlessness. So if there's a lot of stress, it's going to communicate to our osteoblasts to make more bone because it's being stressed. If there's no stress, the osteoclasts then are going to come in and destroy that bone so that bone remodeling can occur. Our bone lining cells, these are going to be the flat cells on the outside surface of the bone where remodeling is not happening, and also on the internal side are the internal surfaces. Here they're called the endosteal cells. Remember, endo means inside. And on the outside, they're called periosteal cells. So peri meaning around. Osteoclasts are going to be derived from the same hematopoietic cells that we see as macrophages. So in a way, they're kind of like a bone macrophage is a good way to explain that. So these cells are going to have ruffled borders that increase its surface area and help to break down the bone matrix. Okay, so if we take a look at the different cell types, we saw the osteoblast, so the bone-making cell. When it matures and it turns into that osteocyte, you can see these little projections here. And what they do is they go into the surrounding rings and they help to sense the stress or the strain. So if the little tentacle picks up on some strain, it's going to tell tell our actively dividing osteoblasts surrounding it that it needs a little bit of help. And then we can see finally the osteoclast, or the bone-making cell, or bone-breaking cell, you can see it has many nuclei, and it's got this ruffled border here as well. So what happens is that kind of acts like a Zamboni, I guess, for an ice skating rink. So it's going to secrete enzymes from here, and then the ruffled border is going to come and shave up that bone and basically tear it down. Okay, so if we see one under a microscope, this is the actual osteoclast right here. So lots of nuclei inside of there, and then you can see here's the ruffled border. So it's going to go across the surface of the bone here and degrade it and kind of eat up that tissue. And here's our osteocyte, so the mature cell sitting in its little lacunae, and you can see those projections coming off from it. Okay. So our compact bone, we sometimes also are going to call that lamellar bone. So what we see in our compact bone are a series of osteons, canals and canaliculi, and interstitial and circumferential lamellae. So remember, our lamellae are kind of like the rings on a tree. Okay, so in an osteon, Okay, it's the structural unit of the compact bone that consists of those cylinders or the lamellae that run parallel to the long axis. So they're going to run in the same direction, and they're going to basically act as like a weight-bearing pillar for the bone. So the osteon cylinder consists of a bunch of different rings, like we said, called lamellae, and within these lamellae they have collagen fibers that are going to run in different directions to help give the bone its toughness and resiliency. So in between these collagen fibers, we're going to find bone salts. So if we zoom in here and we look at this lamellae, we can see the collagen fibers running in one direction, and then the next ring, they're going to run in the opposite direction, and so on and so forth. So what this helps to do is resist any kind of twisting or stretch or strain. And then we can see on the inside of the osteon, or that haversian canal, we've got capillaries, arteries, veins, and nerve fibers. So super important conduit for those materials. Okay, so our central haversian canal is going to run through the core of the osteon. It's going to, again, contain those blood vessels and nerves. And then we also have these Volkmann's canals that are going to come in perpendicular at a right angle to the central haversian canal. So they are lined with endosteum, and they are going to connect the blood vessels and the nerves from the outside of the bone, or the periosteum, into that central canal. So we would have our osteon canal, like a haversian canal here, and these Volkmann's canals are going to come in at a right angle, and it's going to snake blood vessels, nerves, and arteries right up through. Okay, so as mentioned before, we also have the lacunae, which is the home for the osteocytes, or the mature cell, and we also have what are called canaliculi. So these are small little hair-like projections that connect each of the lacunae to one another. So that's where those projections off the osteocyte are going to sit. So they're kind of almost like holding hands with all of the other osteocytes there, so they can stress or sense the strain or the stress. When the matrix hardens, so when it's no longer an actively dividing cell, it's going to get trapped. The osteocyte gets trapped in there. Okay, so couple different lamellae that we have. So the first one is the interstitial lamellae. So these are not part of an osteon. So these are basically just bridging the gaps in between the actual circles. So if we have osteon, get out of here. We have an osteon here. We have one here. They maybe aren't touching, but we've got this interstitial lamellae in between the two. Okay, so if we are looking at the microscopic anatomy of a bone, so see here we have a full osteon. The circumferential lamellae is going to go right around, so they're going to be the outer layer of the bone. And then in between here, these are like the interstitial lamellae in between them. So we can see two things here. We can see our Volkmann's canals, and then you can see these perforating canals at a right angle that are going to go inside the bone. So here's a close-up look under a microscope of what one of these, like what an osteon looks like. So here, this is going to be our osteocyte, so that mature cell that's trapped in its lacunae, and these are the hair-like projections. So see how they touch one another? And that way, if this lacunae is getting stressed, or this osteocyte is sensing stress, it's going to reach out to all these other ones basically for help, and just kind of let them know, hey, we're getting stressed here. I need a little extra help. Okay, so moving on to our spongy bone. It looks kind of like it's unorganized, but it's actually very highly organized along the areas of stress or tension. So it helps our bone resist any stress. Those trabeculae are the little spikes, or like cables on a suspension bridge. So here we don't have any osteons, but the trabeculae do contain some lamellae and osteocytes that are connected by these little holes or canaliculi. So we also have capillaries in the endosteum that supply it with nutrients. So you saw this previously, but these are the trabeculae, or the spiky pieces of bone that we're looking at that kind of act as a suspension bridge that are going to line up along the areas of stress. So when we actually look at the chemical composition of bone, we have both organic and inorganic components. So our organic components are going to be these osteogenic cells or osteoblastocytes, bone-lining cells, class an osteoid. So the osteoid is going to be just that matrix, again, it's secreted by our osteoblasts. So it consists of ground substance and collagen fibers. We know that collagen gives us resiliency, but it also gives us strength. So again, organic compounds are going to give us resilience and have these sacrificial bonds between collagen molecules. So, if they're stressed, they're going to sacrifice themselves and break in order for an area to build up more somewhere else. So they stretch and break to help dissipate this energy and prevent a fracture. I'm sure everyone's been in this situation where you felt like, wow, I really should have broken my bone there, when I fell on my wrist, or I got kicked really hard in my shin. But these bonds allow that not to happen. So if there's no trauma in that area beyond that, these bones are actually going to reform all over again. Okay, so our inorganic compounds are what we call these hydroxyapatites or mineral salts. So they make up about 65% of the bone mass. So this is where that hardness comes in, hardness and resistance to compression. So these are mostly calcium phosphate crystals that we find in and near the collagen fibers. So the bone, interestingly enough, is half as strong as steel in resisting compression and as strong as steel in resisting tension. Okay, so these inorganic compounds are going to last long after our death because of that mineral composition. So this is how we find information about ancient people. They say, well, they ate this, and we know that they did this, and this is entirely how we find that information out. Okay, so just some things to think about. What would happen if you removed all of the inorganic compound from a bone? Would we be able to resist compression or tension? So would we, if we removed these hydroxyapatites, would we be able to resist tension and compression, or what would that actually look like? So you can do this as a lab project. You can take like a chicken bone, or I know sometimes, I think you guys did this in biology, soaking an egg in vinegar for several days, and what happens? Essentially, it's going to dissolve all of those inorganic compounds, and you're just left with collagen, which means that chicken bone that used to be really hard now is going to be rubbery and have zero tension whatsoever. And which cell has a ruffled border and acts to break down bone? I hope you know that is an osteoclast.